on. Thank you. Thank you, organizers. Oops, it's too loud. I think Satya has done an excellent job in introducing hemostasis. What I'm going to tell you, that our organizers have defined this hemostasis as hemostatis. Essentially, they have done an excellent job without knowing. It's an inflammatory reaction. That's exactly what they defined. But hemostasis, as he already pointed out, hemo means the blood, stasis means stopping. It's a very simple mechanism. But when you want to do, as he already pointed out, thrombosis is a complex phenomenon. But Rudolf Virchow has pointed out that there are three important components to it. One is the abnormalities of the blood vessel wall, abnormalities of the blood flow, and abnormalities of what is it called? Blood. Yeah. So all these three components must play together in order for thrombosis to occur. This is what Rudolf Virchow pointed out. But the question is, very recently, genome-wide knockouts and knockdowns have been done in mice to establish the factors involved in a given physiological pathway or given developmental pathway. Done by Elaine Fuchs from University of Chicago. She did mostly the skin genes and identified 2,000 of them play a role in that. We have approximately 25,000. Another work in E. coli, they also established about 300 genes out of 3,000 genes in E. coli for a given specific pathway. So my general consensus is that this pathway is so complex and huge, I predicted 10% of the entire genome must play together. So how do I identify those 2,000 genes? For example, so far in hemostasis, including Satya's pathway, there are about 300 factors known, done by 100 laboratories for the past 60 or 70 years. How do I identify the rest of the 1,700 factors? That is a challenge that we faced in 1995. So I thought that if we can exploit genetics as a tool to use one of these very small fish called zebrafish, which actually hails from India, I would be able to possibly utilize the power of genetics and identify by so-called comprehensive screening the entire 1,700 factors, which at that time was rather primitive. In fact, when I wrote an application to a white elephant called National Institutes of Health, they said, this idea is nonsense. This is very primitive, and therefore you cannot do this work. Fish is primitive compared to humans. But I did not believe them. I believed the genome is so conserved, and fish and ourselves, we diverged away only 450 million years ago, and to create such a complex pathways, 450 million years is just nothing. Therefore, the fish must have a conserved pathway. So then we sequenced all the genes and identified almost all the factors. I'm not going to go to the list of that. That showed the funding agency that fish are important and conserved, except for the other factors that are present here, we still don't know. Now, the zebrafish genome is sequenced. We have 26,000 genes in the genome. So how do we go about one at a time? There is a technique called knockdown technology, which utilizes a compound called morpholinos, which are modified oligonucleotides and stable. These oligonucleotides, they are antisense to mRNA, and if you can just put them together into a system, it will block the production of the protein. So we did that, and we created bleeding for the known genes called prothrombin gene. And then we did also arterial thrombosis. As Satya showed, the so-called traffic jam. Here, exactly a similar traffic jam occurs in the zebrafish vessels. That is the artery where we injure the vessel by laser. And as you can see, all these fish platelets aggregate just as he showed you in the streets of Chennai. So that is called arterial thrombosis. If it takes about 50 seconds to do that, if it prolongs, that is called a knockdown has worked. So we did like that, and in fact, we have shown many genes using this methodology. There it is. Oops, I cannot move this. Yep. So, but the problem is, when I give it to graduate students, they think that it is extremely difficult to do many genes of 26,000. Therefore, they said, we are not going to work in the lab. So I said, there is no choice for me. I invented another methodology called vivo morpholinos. Vivo morpholinos are exactly similar. Look at this. This is previous morpholino I showed you. The vivo morpholinos are exactly identical, but they have got a bulky group here, which will help this morpholino penetrate into the cells, each cell in the body. Suppose I inject this compound into my blood vessel, it will reach the liver, pancreas, everything else except possibly the brain. Therefore, it was easy to inject an adult fish instead of the so-called larvae, and we did that. We showed the proof of principle. Then we thought we should look at the genes. We did microarray and identified 1,000 genes, and we started looking at knocking down each one at a time. But the problem is the cost. 25,000 genes, each gene cost $1,000. So $25 million. Would NIH fund me? Would DST fund me? Absolutely not. Because they're even scratching for $1 million. Therefore, we said an intelligent design of a novel method might do the trick, and we can beat NIH system. And this is called piggyback knockdown methodology, where you take this morpholino, make a regular oligonucleotide, make a complex. This is the group I showed you, the large group, and inject this into the blood. So this portion is complementary to the messenger RNA, therefore it binds. So because it is having that bulky group, it piggybacks the oligonucleotide, standard one, and thus the costs are cut from $1,000 to $5 a gene. So now it is easy for us to do. So I put about 4 graduate students. They have done, we have shown the proof of principle, and then they have also chosen multiple oligos and showed 72 new genes were identified. Remember, for 60 years, 100 laboratories, only 300 factors were found. For a single laboratory, 72 genes were found within a short period of 4 months' time since the technique was invented. So lo and behold, we applied this to another group of genes called HOX genes and a new gene called HOXC11. When you knock it down, you get what is it called? Increase in thrombocyte production, or called thrombocytosis. And in a similar fashion, we have chosen genes which possibly initiate the blood clotting. We have taken about 189 serine proteases, knocked them down, and showed that there is one particular gene called prostacin, which is probably involved in activating the factor 7, which has been an unsolved puzzle for the past few decades. Therefore, in this group, we have discovered many genes, but we chose a few of them and extended their function a little bit. One is called intraflagellar transport protein, which is only present in the flagellum, but which is also present in the platelets now, aryl hydrocarbon receptor, and GPR18. Then the technology of CRISPR-Cas9 came, which is a knockout. We have used this technology and knocked out part 2, factor 8, and also von Willebrand factor. I didn't show you that. So far, that is hemostasis. But then the question remains, how thrombocytes of the nucleus in fish, in what other vertebrates like reptiles, birds, but mammals are the only ones which are insulted to have a non-cellular component called platelets. What is the advantage for them is a question that we raised. The advantage is, if you take a look at thrombocytes and platelets, if you write the properties, they are platelets, thrombocytes. Therefore, It is not like a bee, the beetle, and whatever the other one is. So you cannot just compare morphology and tell that it's possible. So even though I said thrombocytes and platelets are similar, but then we looked at thrombocytes and megakaryocytes, which are precursors for platelets. They also showed several properties which are very similar. It is like comparing a parrot and a carrot. Then we asked a question, when did platelets evolve? During Triassic period, around 240 million years ago, first mammals evolved. That is when the platelets started beginning the process. And then that was hypoxic, no oxygen. Almost all of them had a mass extinction. Those animals which had thrombocytes died, but the platelets one survived. Subsequently, the birds evolved during oxygen increased again. During that time, birds evolved. Birds do carry thrombocytes. They didn't die. The reason is they got rid of the factor that I showed you a question mark in the very first slide. The pathway is unknown in fishes. That the birds got rid of that factor 12 by mutagenesis or whatever. Even diving mammals are hypoxic under the ocean. They also got rid of that particular pathway. So in addition to that, if you calculate the surface area of the platelets, they're extremely large in terms of huge amounts. Thrombocyte surface area is very less. Therefore, they're effective hemostatic agents. Thrombocytes are not as effective as platelets as far as the hemostasis is concerned. But when it comes to the volume, if it fills the lumen of the vessel, thrombocytes, 100 of them will fill the lumen, whereas you need thousands of platelets to fill the lumen. In fact, in our lab, we completely occlude the vessel, but in those labs, people use another animal model called mouse, which is to me is a big fur ball. In that particular animal, it will never occlude under the same conditions, okay? So as far as thrombosis is concerned, thrombocytes are more efficient, and thus the fish and other things would have been killed during hypoxic conditions. I think in summary, platelets are fantastic. I mean, zebrafish are fantastic study genetics of thrombosis. They help in fundamental discovery of platelet clustering. I'm not going to go there. They help in identifying several novel genes, and they help in examining the platelet evolution. And thank you all, and I thank all these colleagues who have worked with me, a lot of postdoctoral fellows, several of them are MDs, and these are some students graduated. This is the current crew, and thank you very much for this talk.